All right, let's return to one of the topics we've talked about previously, which is genetic diversity, but now think about it specifically within the context of conservation genetics. So overall, small populations of species at risk often have low genetic diversity. And so if we think of this hypothetical example where we have a large population, and each of these different colors here represents genetic variance. So the more different colors you have, the more genetic diversity there is. When you take small pools of that from the large population and now we're creating a small population, just by chance, it's more likely that you're going to have lower genetic diversity. That's just based on the way it is, based on the statistics. If you have a small population, they're often going to have low genetic diversity. And that can be for several different reasons. So if we're thinking about, I don't want to write with yellow. So why is that? So why do small populations have low genetic diversity? First of all, we think about which process has more of an effect in small populations. Genetic drift is more likely to have a strong effect. If genetic drift acts more rapidly in these small populations, that can lead to fixation of these different alleles and depletion of genetic diversity. Another reason is that if we think about these two different small populations, they're often isolated, which means that gene flow isn't occurring between them. So if gene flow is unlikely, gene flow is one of the processes that increases genetic diversity. So small populations often have low diversity. And then lastly, small populations are also more likely to experience inbreeding, which is something we will dedicate the entire next video to. But these are some of the reasons why small populations often have low genetic diversity. And that low diversity can have some serious consequences. So two possible consequences of low genetic diversity are loss of evolutionary potential, which is what we will focus on in this video, and also inbreeding, which is another very important negative consequence, but we'll focus on that topic in the next video. So low genetic diversity overall decreases the likelihood that some members of a population will have the necessary underlying genotypes to adapt to new environmental conditions. So if we think about that example where we had all the different colors in that circle, if you have a lot of different possible colors, it's just statistically more likely that one of those genotypes, one of those colors, is going to be able to adapt to these new conditions. But if you have low genetic diversity and a low number of genotypes, it's less likely that one of those available genotypes will be able to have the variation needed to adapt to these new conditions. So they've done some really interesting studies with this, cited here. This is a type of crustacean where they manipulated in the lab two different populations. So they had one population that had low genetic diversity and one population that had high genetic diversity. So I'll use GD to abbreviate genetic diversity. And what they found in their study is that if a population had low genetic diversity, they also had low fitness, and they actually ended up going extinct when the conditions that they were being housed in became stressful. In contrast, if they had high genetic diversity, they were actually able to survive those stressful conditions. So this is one example of how high genetic diversity can help a population survive in new stressful environmental conditions. Again, this study was done in the lab. It can be harder to figure out the evolutionary potential of natural populations, but we'll talk about a couple different ways you can do this using molecular data. So we'll go through in this video. You can identify adaptive genes. You can quantify standing genetic variation. can identify correlations with population size. So those are the three ways you can identify evolutionary potential in natural populations using genetic data, and we'll talk about each of those in the rest of the video. So let's start with adaptive genes. So adaptive genes can be used to identify or infer the evolutionary potential of a natural population. So if we think back to this plot that I keep showing you where we have FST and heterozygosity, we have a bunch of different dots, and then we have a few SNPs that are what we call those outliers in that red region. Those are the adaptive markers. Those are the outliers, right? So what we can do is identify those adaptive genes and then using techniques like BLAST, compare them to a known database to figure out what they are. The problem that your textbook mentions is that oftentimes what we end up finding are what they call anonymous adaptive genes, where when you end up comparing them to the database and blasting them, it actually doesn't find any significant similarities, and so you don't know what that gene actually is. You know based on its FST and heterozygosity values that it's an outlier, but you can't actually match it to the function. So from a conservation management perspective, that's problematic because really this approach is most informative if we know the functions of that adaptive gene. So an example of that is in the Tasmanian devil, which recently has been suffering from a fatal cancer, and in the past 2 decades it's declined more than 80%, something really dramatic like that. But because this provides such a strong selection pressure, they've actually found in a couple different populations different genetic regions that are under positive selection and are actually related to functions that make sense, like cancer risk, immune regulation. So that's what's shown here in this table, are all of the adaptive outlier genes that were found to be under selection in these populations. They're all on chromosome 3. And here on the very far right-hand column is the putative functions, again, mostly related to cancer and immunity. And so they think that there's actually selective pressure now where some of these individuals might actually be resistant to this cancer. The few that had the genetic adaptive variation available to allow them to survive are the ones that are surviving. They're the ones reproducing. And so they're trying to use this information for potential reintroduction programs. And so when you're doing that, you want to make sure you're identifying individuals that have these adaptive variants that will help them be more likely to be able to survive, and it's more beneficial to have these resistant genotypes. So this is one example of identifying adaptive genes and how you can use that to particularly choose which individuals you want to use for reintroductions, which we'll talk more about later. Another technique you can use is identify standing genetic variation. And standing genetic variation can be used to infer the evolutionary potential. Standing genetic variation, again, if we go back to this hypothetical example, the more colors you have in here, the more standing genetic variation you have. So standing genetic variation basically just means a lot of different potential genotypes. So they found in this example cited here, and this can also relate to invasive species and founder effects and bottlenecks. So remember, we've talked about this previously in terms of genetic diversity. When you have an invasive species, often you have a founder effect happening where only a few individuals are the ones that are doing the invading, and so very low genetic diversity is contained within those new individuals. And so they've actually found that here with this particular European plant. So initially that species was found down here in the bottom right. This is the core population. And then it expanded into several different regions in the Mediterranean, for instance, over in Spain and Portugal. And what they did is compare the genetic diversity and standing genetic variation in these different populations. And they found that because this founder effect happened, these what are called range edge populations, where these are the ones that have expanded out, these populations actually have low, I'll call this standing genetic variation. These populations have low standing genetic variation because of that initial founder effect. So these populations are less likely to be able to adapt to changing environments, whereas this core population has higher standing genetic variation and they have a better chance of being able to adapt. Another way you can use techniques we talked about previously to try to infer evolutionary potential is to identify correlations with effective population size. So remember, in general, if you have a small population size, drift will have a larger effect and selection will have a small effect, and vice versa. If you have a larger effective population size, drift will have a smaller effect and selection will have a stronger effect. And selection is that process that can help cause evolutionary change that's beneficial. So what you can do is look for correlations where if you have a larger effective population size, which is shown here on the x-axis, you would also expect to have higher evolutionary potential. So this metric here, it's a little bit convoluted. I looked into this paper, but it does relate to some of those DN-DS ratios we talked about previously with non-synonymous and synonymous substitutions. But overall, the higher value here on this y-axis indicates higher evolutionary potential. And we can see in this paper across several different groups of organisms, overall there's a general positive correlation here where the higher the effective population size is, the higher the evolutionary potential is. So we can use that metric of effective population size to help us determine whether this population is likely to have high or low evolutionary potential. So those things we've talked about are all related to the genetic diversity in terms of the actual DNA sequence and the nucleotides that are present in the DNA. So genetic diversity absolutely affects the conservation of species. But it's also important to remember that gene expression can also play a role. So there are several different examples of this, even if the DNA is exactly the same, if the transcription and the RNA expression is different, that can also lead to differences. So remember we talked about previously that example with the different subspecies of crows, and one was all black, one had the white hood around its neck. The DNA sequence was identical, but their gene expression was different, and that led to these phenotype differences. So when we're thinking about conservation, it's also important to remember that gene expression can also play a role in terms of how adaptive one population might be in terms of thinking whether it'll be able to survive long term in its habitat. So gene expression is one thing to consider, and epigenetic modifications can also affect a species' success. So I think we talked about this way back when at the beginning of the semester, but epigenetic basically means above the genome. So in this picture here on the left, often when we talk about epigenetics, we're talking about the addition of these methyl groups. So it's not affecting the DNA sequence itself, but it's adding these things to the DNA that can then oftentimes affect the gene expression. And so a really cool example of this is in the Japanese knotweed, which is shown here on the right, which is a plant that was native to Asia and has now invaded a lot of North America. And what's really interesting is that this plant is actually, in many cases, clonal, meaning the DNA, the genotypes are identical. So we might expect that plant to not do very well then if it has basically no genetic diversity whatsoever. But what they found is that there are actually epigenetic modifications in this plant, so you can have the same clonal genotype, but different epigenetic modifications potentially allow it to inhabit these different types of environments, and so it can respond differently and adapt differently to these environments, not because of its genetic diversity, but because of its epigenetic modifications, which I actually didn't know before and is super cool. And lastly, just to, again, tie all of this together to what we talked about previously, when we're thinking about the best way to identify whether populations have the potential to evolve, whether they have these evolutionarily important genes, really the best thing we can do if we have the resources, is to look at variation across the entire genome and to do that whole genome sequencing. That's because oftentimes those adaptive genes are so few and far between that they'll be easy to miss if you don't sequence the whole genome. So really, it's best if we look at everything, and then we have a better chance of finding those genes that can be important and informative in terms of helping us determine how that population will be able to fare in these changing environments.